Hello everyone, I am representing the team the last six brain cells and we are here to present our solution for problem statement ID 25042, identifying taxonomy and assessing biodiversity from eDNA datasets. Our project is called GeeKhoj AI, an AI discovery engine for marine biodiversity. The current existing methods fail to identify over 50% of deep environmental DNA, simply because it belongs to unknown species. Unexplored data is often discarded. That's why we have designed GeeKhoj AI to provide an end-to-end AI pipeline that processes 100% of raw genetic data. Instead of discarding unknown DNA, it uses both supervised and unsupervised learning to classify known species and discover novel organisms. This way, we create the first ever complete census of marine biodiversity, covering both known and unknown life types. So, welcome to BioDash, our working prototype of GeeKhoj AI. In this section, I'll walk you through how it functions in real time, from dashboard to data flow, insights, and visualization. From the first section, that is environmental DNA, eDNA analysis and sampling. We see that details of samples are collected, processed, sequenced, and prepared for AI classification. This includes field sampling, that is collection of water samples from the environment, DNA extraction and purification, high throughput sequencing, bioinformatics pre-processing. Basically, let us understand how raw DNA is generated before being fed into the AI pipeline. Moving on to taxonomy and biodiversity, basically species classification. Here you'll see the taxonomy tree, which includes the kingdom, phylum, class, order, family, genus, species of the organisms that are identified in the dataset. It also provides biodiversity metrics, that is, the richness, how many distinct species exist, the evenness, abundance of weeds per taxon, etc. The section will allow a researcher to see what life is present and what proportion of data has been classified or unclassified. Navigating to meta barcode workflow, this section explains how the AI works. Basically, feature extraction from DNA sequences, converting the raw reads to features usable by ML or AI. The supervised classification part includes the genomic transformer or a similar model known for taxonomic assignment. The unsupervised discovery part is just clustering or anomaly detection on sequences that don't match known species. It includes different types of bioinformatics and taxonomy as well. Moving on to the deep sea challenges section. Here, users can see the abyssal plates or the hypothermal vents, the sea mounts that the organisms are belonging to, or the extreme environmental conditions that they are living in. Moving on to our data repositories that are being used. This includes curated marine taxonomic sequences, clean and validated datasets, perhaps databases of barcodes that are known, and the proportion that is still unassigned. This is very crucial because classification depends on having a reference to compare against. All in all, once data is processed, users can filter the results using environmental metadata, that is the sampling location, the depth, time, etc. Visualize taxonomy via interactive tree diagrams to see which species or groups are present. Compare biodiversity metrics across samples or sites. Explore clusters on unassigned sequences to see potential novel organisms. If the sequence includes an explainable AI, that is XAI components, we show saliency or highlighted features that indicate why a sequence was classified in a certain way or flagged as novel. So now you might wonder what makes our AI unique? Well, it is a hybrid AI engine that we are going to be using. A supervised genomic LLM classifies known organisms, while an unsupervised discovery engine processes the rest to uncover novel life. The anomaly powered discovery, our autoencoder AI detects DNA that doesn't match known species and flags it as new. And beyond identification, we don't just provide a species list. Our system predicts each organism's ecological role, giving a true picture of ecosystem function. Now let me walk you through what exactly is the data flow in our system. Firstly, raw eDNA files are uploaded in FASTQ format. Then genetic features are extracted for analysis. The system, that is our AI triage, will separate known from the unknown DNA. Now, part A would be supervised classification that includes known DNA sequences, which are identified using a genomic transformer. And part B, that is our unsupervised clustering, where unknown DNA is grouped into potential novel species. Finally, we will use our AI to assess the ecological role of both known and newly discovered organisms. Researchers interact with this process through a user-friendly dashboard, which includes uploading DNA sequences, attach environmental metadata, and launch new analysis jobs. Track real-time progress, view biodiversity metrics, visualize taxonomy, and explore insights through interactive dashboards. The main challenges that we'll be facing include collecting and cleaning our large data sets, running genomic models on GPUs, and AI-led novel species, which must be verified by experienced biologists. To overcome this, we use transfer learning, cloud GPUs, and collaborations with marine research organizations like the CNLRE. To conclude, our solution, Jeep Khoj AI, is not just a classifier, it's a discovery engine for marine life. By ensuring that no DNA data is wasted, we take a major step toward uncovering the mysteries of the ocean and safeguarding